Hello, Year 10s. We're on the health and disease topic towards the end. You should have most recently have looked at how vaccines work and how the immune system helps to defend our body once the pathogen gets inside the body. But today we're going to focus on antibiotics. When do we use them? How do they work? And how do they get into production? So wherever you see the shape, that's where you need to stop the video and follow the instructions. Now, these are the outcomes of this lesson, but what I want you to focus on is just think back for a moment on the ways in which our body tries to protect us from harmful pathogens. Pause the video, have a think. Okay, right. What I've done over here is I put a summary together of different ways in which our body stops pathogens. And what you could do is you could go to the PDF of this video and you can print this page or you can copy it into your book. So the first thing we know is that our body has physical and chemical barriers to stop the pathogens from getting into our systems in the first place. And some of those physical chemical barriers can include the skin. It could include the mucus and ciliated cells in our respiratory system that trap dust and pathogens and then waft it out so we can sneeze it or cough it out, or rather disgusting. Earwax again traps any pathogens. Lysozymes. Now remember, these are special enzymes found particularly in our tears that help to kill any pathogens that might try to get in through our eyes. Scabs are caused by a particular type of blood cell called platelets, which cause a fiber to be formed and stops the blood cells from going through and eventually forms that scab to stop any new pathogens from entering where that poorly occurred. And of course, there's that wonderful hydrochloric acid in our stomach. If there's any food that we've ingested, such as a badly cooked sausage at a braai or a barbecue, the hydrochloric acid in our stomach will try and destroy that pathogen. So apart from those physical barriers, we also have the immune system to help us once it's got through those barriers. So the immune system is all about stopping pathogens once they've actually got in. And the different parts of our immune system really related to our white blood cells in our blood. One type of white blood cells called a phagocyte, and remember they engulf the pathogen and then they literally ingest it, they gobble it up, or they get other white blood cells to kill the pathogen. And then we get lymphocytes. Now, lymphocytes is a type of, the B lymphocytes, a type of white blood cell, and their main job is to produce antibodies. Remember, the antibodies match the antigen on the pathogen. This is a very specific shape, so it figures out which antibodies are needed, which white blood cells have those antibodies, and then they produce loads and loads of antibodies to kill the pathogens. We also, at the bottom there, we have the killer T cells. They actually destroy the pathogen. And then we have the memory T lymphocytes. The memory cells, they remember the response that's needed for that particular pathogen, like the COVID virus. And if you are infected again, they will respond far quicker with a much higher concentration of antibodies so that the pathogen is destroyed before you feel any symptoms or realize it's ever entered your body. And remember, a vaccine is a dead or weakened version of the pathogen to bring about this immune response to build immunity. And the reason why the COVID virus vaccination we have two is because the second one, the level of antibodies, the level of memory cells in our body goes up, making us immune for a longer period of time. So I hope this has helped to summarize that. Now, and I didn't, there remains in the blood in case the pathogen comes back. Now, either pause, copy this down, or print it, but make sure this goes in your book. Okay, so let's try a bit of a quiz. Which type of blood cell is important in the immune response? pause, think of your answer, and then check. Of course, it is white blood cells, or if we want to be more scientific, we can say lymphocytes. What name is given to the molecules and pathogens that the immune system responds to? Remember, it's on the pathogen, so we call it the antigen. Well done. How about this one? Which cells are left in the blood after an infection has been cleared? Well done, you've remembered. It's, of course, the memory cells or the memory lymphocytes. Give 2 ways that a secondary response differs from a primary response. So this is about the first time you get the infection or the vaccination, and then the second time. Of course, the second time, the response is a lot faster and a lot more antibodies are produced. And think about that graph that you were shown in class. Which name is given to the inactive form of a pathogen used in immunization? Of course, it is the vaccine. Well done. Which medicines are used to treat infections caused by bacteria? So you go to the doctor, he's diagnosed you to have a bacterial infection of some sort. What kind of medication does he give you? Okay, really important. It's antibiotics. We use antibiotics against bacterial infections. When possible new medicines are being developed, what are they first tested on? Okay, very often they test on cells or tissue cultures, and then they are tested on animals. Now, just listen to this. You don't need to write this down, but this guy is so important. Alexander Fleming, a real hero in the biology world. In 1928, he noticed that on some agar that he was growing bacteria on, there was a mold developing, a type of fungus. And what he noticed was where this fungus was developing, the bacteria had actually died. There were circles of clear areas where the bacteria had died. And this is how he discovered that actually that mold was killing the bacteria. And that was brilliant. And actually it turned out that the name of that mold was in fact penicillin. Now I know some people are allergic to penicillin, but penicillin is an example of a antibiotic, and in fact it grows on our bread mold. An incredible discovery that was a real game changer in the medical world. Right, so make sure that you understand the word penicillin, an example of an antibiotic. Fleming, that you can attribute him to discovering antibiotics. The mold being the fungus, and of course bacteria, which is what the antibiotic kills. Now what I'd like you to do is pause this video and watch this really great video on YouTube about what antibiotics are and how they work, and then come back to this position on the video. If it doesn't work in Chrome, open it up in Internet Explorer. Okay, now after watching that video, I hope you can complete this sentence. So this is these few sentences. There are some gaps, and what I want you to do is underline the words that you fill in. So pause and copy these few sentences in your book, and then you can purple pen your answers. Okay, so here we go. Here are the answers. Antibiotics are substances which either kill or inhibit cell processes of the bacteria. This stops them growing and reproducing. Antibiotics do not have this effect on cells, making them very useless. Human cells, making them very useless for viral infections. So often they ask a trick question in the exam. If you go to the doctor and he diagnoses you as having flu, why does he not give you antibiotics? And the reason is because antibiotics only work for bacteria. Remember to put the two B's together. So make sure you've copied these notes down fully, because these are the most important notes you're going to get on antibiotics. And then for the last couple of minutes, we're just going to focus on a topic that we looked at a little bit earlier, on antibiotic resistance. Okay, now we know that in a population of bacteria, Some bacteria are going to be varied. So just like all of you, if you got into the swimming pool, some of you are going to be much faster than others. Some of you are going to go, I don't feel like swimming. Some of you are going to go, the water's too cold. All of you are going to react differently to the situation. And the same thing happens with bacteria. There is variation in the population. Now, if you're given a course of antibiotics, with time, more and more bacteria are killed. And that's what we expect. But some bacteria don't die straight away. And if we don't follow our full course of the antibiotics, so if the doctor says take it for 7 days, and after 5 days you go, oh, I'm feeling better, I'm not going to worry about taking them anymore, and you stop taking, then these stronger, or what we call resistant bacteria, they start to reproduce and multiply. And it can happen that at a point in time, those antibiotics won't react or won't kill the bacteria at all. And MRSA is an example of a bacteria where exactly this happens, and it means that a simple bacterial infection that would normally be killed is very, very difficult to treat, and we call these superbugs. So it's really important that you make this connection with natural selection as you put this together. Okay, now in the next video, I'm going to have a look at how drugs and vaccines eventually get out in the public after they've been very, very carefully tested. So good work, well done, and look forward to seeing you in the next video. Don't forget to subscribe to my channel. I'm really trying really hard to beat Mr. Gregson with the number of subscribers he has.